Assalamu alaikum. So today we'll be discussing about the heme degradation. So what is heme? Basically, heme is the synthetic part of hemoglobin. Hemoglobin is from heme, and combination with globin. Heme is synthetic part, prostatic part, and globin is the protein. So this is how when heme is combined with globin, it forms hemoglobin. Then hemoglobin is introduced in red blood cells in the erythropoietic system, and then this is how the hemoglobin is basically entered into RBC, and the matured RBC contains hemoglobin which carries oxygen from one place to another, specifically from the lungs to the tissues. When its work is done, or when the RBCs have grown old, like the lifespan is 120 days, so RBCs are broken down specifically in spleen. So that's why the spleen is also known as the graveyard of red blood cells. Hemoglobin is degraded, and hemoglobin is again broken down into heme and globin. Globin is basically stored and reused, or sometimes it's degraded to form different kinds of amino acids so that we can make different kinds of proteins from them. Heme is degraded. Heme is formed from two components, protoporphyrin rings and iron. Iron is basically extracted from the heme, and it is stored for the reuse. Iron is never excreted from the body, and the heme, the remaining protoporphyrin part, is basically converted to biliverdin and then to bilirubin and then excreted from the body. So erythrocytes becomes old, lose their flexibility, become spherical in shape, rigid and fragile. Once the cell becomes fragile, they are easily destructed during the passage of their tight circulation spots, especially in spleen. So that's why the spleen is known as the graveyard. The intracapillary size of spleen is around 3 microns as compared to the 8 micron cells of the red blood cells. RBCs are basically have a lifespan of 100 to 120 days, after which they become trapped and fragmented, and they have to be broken down. For this, your spleen is there. During erythrocytes are engulfed and destroyed by macrophages. So red blood cells is broken down to red blood cells released hemoglobin. Hemoglobin is broken down to globin, which is either reused or converted to amino acids, broken down to amino acids, and amino acids go into amino acid pool to make different kinds of proteins. The heme part, iron is taken out and recycled, reused. Heme is converted, the protoporphyrin rings are converted to bilirubin, which is excreted from the body. The globin is recycled. Heme oxidizes when the heme porphyrin ring becomes opened by the endoplasmic reticulum enzymes known as heme oxygenase. So heme is basically when the iron is taken out from it, the porphyrin rings is acted upon by heme oxygenase, which is present in the endoplasmic reticulum. The oxidation occurs in specific carbon producing equimolar amounts of biliverdin, iron and carbon monoxide. This is the only reaction in the body that is known to produce carbon monoxide. So the carbon dioxide is also produced, and carbon monoxide is also produced in our body. Most of the carbon monoxide is excreted through lungs, with the results of that the carbon monoxide content of expired air is a direct measurement of activity of heme oxygenase in an individual. So if we want to check that the heme oxygenase is working or not, we can check the amount of carbon monoxide exhaled in the exhaled out from the lungs, exhaled air from the lungs. This is the heme containing iron and 4 porphyrin rings, protoporphyrin rings. Then heme acted upon by heme oxygenase, oxygen and NADPH which is produced in HMP shunt. Iron is released, carbon dioxide, water and NDP is released. Heme is oxidized to biliverdin. 1, 2, 3, 4. These are the 4 porphyrin rings which are made, which makes biliverdin. In the first reaction, a bridging methylene group is cleaved by heme oxygenase to form linear biliverdin from cyclic heme. The cyclic form is basically converted to linear form of the biliverdin. Iron is released from the ring in the process. Second reaction, when these biliverdin is acted upon by biliverdin reductase enzyme, again NADPH is utilized, and biliverdin is reduced to bilirubin. ठीक है. Another methylene bridge is between 3 and 4 is reduced biliverdin reductase producing bilirubin. ठीक है. This is the methylene between 3 and 4. So two important enzymes are important. Heme oxygenase, which converts protoporphyrin rings of the remaining heme into biliverdin, and biliverdin is again acted upon by biliverdin reductase enzyme, which converts into biliverdin into bilirubin. Bilirubin is an orange yellow pigment derived from censored red blood cells. It's a toxic waste produced in the body, so it has to be excreted from the body. We cannot store bilirubin. Extracted and biotransferred mainly in the liver, excreted in bile in urine in the form of urobilinogen and stercobilinogen. It's a bile pigment. It is basically released in the bile or carried by the bile to the second part of the duodenum. Elevation in serum, blood and urine bilirubin levels are normally associated with jaundice. When the bilirubin is increased in the blood, it is labeled as jaundice. So an average person produces about 4 milligrams per kg of bilirubin per day. The daily bilirubin production from all sources in man averages around 250 to 300 milligram. Hemoglobin from censored red blood cells destroyed in endothelial system, liver and bone marrow, and around 85%, 15% destroyed in the bone marrow, or catabolism heme containing proteins like myoglobin, cytochrome and peroxidases. So 15% bilirubin comes from these things and 85% from the hemoglobin. In total 250 to 300 milligrams per day of bilirubin is produced inside a man. Biliverdin causes a change in yellow color of molecule from blue-green biliverdin to yellow-red bilirubin. The later catabolic changes in the structure of tetrapoles are responsible for progressive change in the color of hematoma or bruise, in which the damaged tissue changes color from initial dark blue to red yellow and finally to yellow color before all the pigments transported out of the tissues. This is how when you get an injury or damage, there is a bruise which basically changes color from days to days, and that's how the dark blue, red yellow and a yellow color is formed. That's due to the destruction of red blood cells and release of the biliverdin and bilirubin. Peripherally arising bilirubin is transported to liver in association with albumin, where the remaining catabolic reactions takes place. So bilirubin is basically insoluble in water, and it has to be carried with some thing which can transport it to the liver, because the conjugation of liver or the excretion of conjugation of bilirubin or excretion of bilirubin takes place in the liver. So liver is responsible for that. To all of the bilirubin which is produced anywhere in the body has to be transported to liver and is transported binded to the albumin. Heme, this is the reticuloendothelial system. Heme is acted upon by heme oxygenase. It's converted to biliverdin. Biliverdin is acted upon by reductase to form bilirubin. Bilirubin is basically compound or complexed with albumin and brought to liver. Then this bilirubin is conjugated with UDP glucuronate to form bilirubin diglucuronate, which is released or excreted through bile into the second part of the duodenum. So this is system which basically is the degradation of heme. Heme is degraded to biliverdin. Biliverdin is converted to bilirubin. Bilirubin is attached to albumin, which transports it from anywhere in the body to liver. Liver then conjugate it with UDP glucuronate. And why we need to conjugate it? Because bilirubin is insoluble in water and it will not be excreted in the bile. So we have to make it soluble, and to make it soluble we have to conjugate it with glucuronate. So bilirubin diglucuronide is basically water soluble, and it basically is transported through bile into the second part of duodenum in the intestines. In the cells of liver, bilirubin undergoes modification to increase its water solubility, so it can be excreted more easily. Two molecules of glucuronic acid creating bilirubin diglucuronide is transported out of the hepatocytes into the bile caniculi and is thus excreted in the bile. The bilirubin synthesized in spleen, liver and bone marrow is unconjugated. It is hydrophobic in nature, so it's transported to liver as a complex with albumin. Okay. This is how it is done. In the microsome of ER, conjugated bilirubin is converted to water-soluble mono or di conjugates by sequential covalent coupling with glucuronic acid. Two UDP molecules of glucuronic acid, UDP is taken out, and glucuronic acid attached to bilirubin, and this is how the bilirubin diglucuronide is formed. This is liver. Basically these are the caniclies, and they form the they basically store this bile into the gallbladder, and the gallbladder this bile duct and pancreatic duct makes common bile duct, which is opened in the second part of duodenum. Intestines conjugated bilirubin are poorly reabsorbed but are partially hydrolyzed back to unconjugated bilirubin by catalytic action of bacterial beta-glucuronidases. In the distal ileum and colon, anaerobic flora mediate further catabolism of bile pigments. Hydrolysis of conjugated bilirubin to unconjugated by the bacterial glucuronidases, multi-steps hydrogenation reduction of unconjugated bilirubin to form colorless urobilinogens, and oxidation of unconjugated bilirubin to brown colored mesobilfiscins. Urobilinogens are collectively termed for three groups: tetrapoles, stercobilin, mesobilin, and urobilinogen. Up to 20% of urobilinogen is reabsorbed from the intestine and enters the enterohepatic circulation. Most of the reabsorbed urobilin is taken up by the liver and excreted in bile. Small fraction enters the general circulation and appears in the urine. It goes into the blood and then is excreted in the urine. In the lower intestinal tract, the three urobilinogens spontaneously oxidize to produce the corresponding bile pigments: stercobilin, mesobilin, and urobilin. They are excreted in the feces, and this basically orange brown color are the major part of stool, and that gives the clay or brown color to the stool or the feces. These stercobilin, mesobilin, and urobilin, which are the compounds of urobilinogen. Urobilinogen are those which are converted in from the bilirubin diglucuronide. The little amount of this urobilinogen, which is reabsorbed in the hepatic circulation and then given to blood, it converts to urobilinogen, which is excreted in the urine. So in the urine and the stool feces both have the bilirubin pigments. So that's with the degradation of heme.